Okay, so today I'm going to walk you through in the next couple of hours about what a drug is, how they came to be, and some of the principal terms that you'll hear about drugs and when they're, and then this relates to how they're used. So we're going to go over the historical use of bioactives. Humans have been using compounds that come from outside the body to treat different conditions for a long time. And this sort of came to be in a more formalized way in the 20th century and creates all the agencies that you know about and hear about. And then we go into trying to define what do we mean by a drug? And what is a drug acting on? What is the target of the drug? Concepts of agonists and antagonists. So some drugs are acting as a replacement for something you don't have, or they could be acting as a compound that's going to prevent the activity of the compound that you normally have, but that process is sort of out of control. And then we're going to talk about whenever you administer a compound to a human, any one of you, any of the drugs that we might take for any reason, there is an effective dose. There is a dose that isn't big enough, and that can vary from person to person. So one pill may not be sufficient for you, but it might be sufficient for somebody else. So small people you might think will need less drug than bigger people. And then if you take too many pills, when you get a particular drug from the pharmacy, they will usually come with instructions of exactly how you can use it. But if you take too much of it, eventually you will get to a point of toxicity. So pharmacology is always aligned with toxicity. And you hear about this when new drugs come to the market and they've been studied maybe in 1,000 people, but when it goes out to the general population, then it becomes evident that there are people out there who are much more sensitive to the drug, and it can cause serious side effects, including death. So we have to be very, very careful when we use the drugs. So that's the big picture, and there's a guy called Paracelsus, we'll get to him in a minute. Okay, so humans, before we were in any kind of civilization, lived like other animals, and we didn't have pharmacies to go to. There was no organized way of understanding things. But general knowledge began to have impact on what people did. So you, for instance, you were out there and there's a particular type of mushroom, and it killed the person last week. You're not going to eat that mushroom today because that's got a toxin in it. So the people initially, of course, it was just designed for those who had the best enzymes for getting rid of the toxin, to metabolize it into inactive forms. And so the people who had the best enzymes were the ones who survived when they ate something perhaps they shouldn't have. And then we learned to use cuisine, the use of fire. So there were foods which were quite toxic, and we still have them. We probably cook a lot of things today which you are cooking in the first place to make them edible and non-toxic. So if you ate them raw, they would be nasty. But if you cook them, then they become perfectly good for human consumption and combining foods. And then as we got more organized, going back about 10,000 years, the concept of agriculture came in, whereby wherever people were living in the world, that they could domesticate the plants that were endemic to that region, and could make and grow the plants in such a way to make them have the best properties, grow faster, give bigger beans, give more beans, whatever. So people became systematic in the use of foods. And that enabled the expansion of the human being, because we were very much limited by the food supply that was available to us. And there's a great book, if any of you have time to read it. It was a Pulitzer Prize-winning book in 1998 by Jared Diamond, who's at UCLA, trying to understand how this food culture and utilization of plants and animals, how this was different in different parts of the world, and how this affected the onset of civilization. It was a very interesting book. And nowadays we have to think of another thing because of the invention of the aeroplane, of the train, of the car. People who had grown up over many, many, many generations in a particular part of the world had adapted to the food and the temperature and the sunlight of that particular region. But now, with all these modes of transportation, we can move these people to almost anywhere. And there is a consequence, and we'll get to that in a little minute. One of the classic, and probably the earliest clinical trial that I know of, concerns the voyages of Captain Cook, who went from England as part of the exploring that was being done in those times into the South Pacific, and it meant that they were on ships for a very long time, away from any sources of fresh food. And so scurvy, which is a vitamin C deficiency, was very prominent, and therefore the sailors on board these ships got very sick. So the Admiralty, seeing this not only in Captain Cook's voyages, but other long voyages, decided they had to systematically investigate this. And so a Scottish physician called Minn selected 12 men from the Cook ship that was suffering from scurvy, divided them into 6 pairs, and gave each pair different supplements. Now, this is not a particularly big clinical trial. These days, we would have much larger numbers to ensure that this was statistically relevant. So the various things that were tried was a quart of cider a day. Well, that sounds perfectly good. I could deal with that. I'm not too sure I would like 25 drops of sulfuric acid 3 times a day on an empty stomach. Now, of course, the stomach is quite acidic. It's hydrochloric acid in our stomach. I'm not too sure that the sulfuric acid going down would be a very nice experience. Half a pint of seawater. I didn't think it was supposed to drink seawater. And then a mixture of garlic, mustard, and horseradish. And this is sort of interesting because all of these compounds contain isothiocyanates. And although it's well beyond this class, this is something that we are actively researching, is the role of the isothiocyanates in controlling the expression of genes. So when you eat your horseradish, the tangy taste that comes from it, that's the isothiocyanates. 2 spoonfuls of vinegar each day, and then finally they tried some citrus fruits. And of course it was the citrus fruits that worked, and only 1 or 2 people got scurvy on the second voyage when they took with them citrus fruits. And this is allegedly where the term liming comes for an Englishman, is that they took limes. The limes were particularly thick-skinned and survived very long periods of time on the ship. So they found that there were compounds in the foods that had bioactivity. And of course, the concept of vitamins wasn't really properly established until the late 19th century and early 20th century. So they didn't know what was doing this. They had no idea of the chemistry of what was going on, but they knew it worked. So I mentioned about travel just now. So people can move out of the niche they grew up in. So people like me emigrated to Australia, found a bonsai beach, said, "Way, this is great, get a suntan," and unfortunately got skin cancer to follow. So that wasn't very clever. The people who put on board slave ships to bring from Africa to the USA went through horrible conditions on the ship. They had terrible dehydration. So the ones that actually could retain sodium and water were the survivors. Well, unfortunately, the descendants of people who now have hypertension. And then one that I was involved in about 10 years ago was the issue of the people who came to England from India, from East Africa, to live in London. Now, London, compared to Africa and India, has very little sunshine. And so if you already have a skin that's quite dark, that's going to prevent sunlight getting through into the skin, it can be very hard to make vitamin D. So there's a real problem with people who, from those countries, who move to England and getting enough sunlight in order to make vitamin D. There's a very similar group in the US. It's the people who came from Somalia and Ethiopia as refugees who the immigration service decided should live in Minneapolis. And you can imagine in the middle of winter in Minneapolis, you're not going to go outside and get the sun in your skin. It's more likely to freeze your arm off than, so it's a real problem. If these people don't take supplements to their diet, then they're going to get osteoporosis. Okay, so bioactives have been used in all sorts of ways. In closed societies, they've been used by the shaman to put the shaman into trances. In the trances, they think wonderful things that they wouldn't think when they were not in the trance. And they can be used to put on the tips of spears and other ceremonies. People use leeches. This goes back in several hundred years that leeches were used as a way of cleaning the blood. And now we realize with modern science that there are actually compounds within the leech that can be isolated, which are very important for preventing blood clotting. And there were many plants that were out there that were bioactive and that people knew was part of these earlier societies. I mentioned before about dose. So dose is really important. And so if you take enough of the compound, however it's derived, whether it's made synthetically, or whether it's in one of these plant extracts, there comes a point where it works for you. And then if you keep adding it, eventually it becomes toxic. And so this is a philosopher from the 1500s, Paracelsus, who said that basically everything is toxic. It's just a matter of dose. So we'll get to this later when we talk about therapeutic index. The therapeutic index is the range of concentrations that you can safely give to humans. And so pharmacologists and clinical pharmacologists try to work out what this is. So it's got to be a dose that's big enough to get the physiological effect you're anticipating, getting rid of your headache maybe, but not so big that it causes toxicity, which could come in all sorts of forms. It could be diarrhea, nausea, but it could be more serious. It could be as much as death. So everything eventually is toxic. Anyway, so some years ago, Dr. Marquez, who was just about to retire as Vice President for Research at UAB, called me up on a Saturday evening. It's always a little nerve-wracking when a Vice President of the university calls you at home. He's not quite sure what's going to follow. But it turned out his wife is the director of the Museum of Art here in Birmingham, and she had a visitor. And the visitor was going to give a talk about the use of drugs in Amazonian societies. And he wanted to tell me I should go and listen to him because the guy was really fascinating. So another professor, Richard Salters, was down in Amazonia during the 1930s and eventually became very, very famous for his work on some of the other phytoactive compounds. But he noticed that the shaman there were using a particular plant to put themselves into a trance. And also, they were also using other things as medicines, and of course as poisons, as I mentioned before. You could put nasty things on the tips of your spear that you were throwing at somebody. And they're still doing this. And the person who came to talk at the Museum of Art was Jeremy Narby. He was a Berkeley PhD student, and he went down and lived in Amazonia for a couple of years and wrote a very wonderful book on it. And what he noted was that this painting up here is a painting of somebody who is trying to represent what is seen in the minds of the shaman during the period when they're in a trance. And if you notice, it's full of twisted snakes. It's very interesting culturally, to compare that to what we believe in as the basis for medicine today, which of course is also a form of the twisted snake, except that we call it DNA. So conceptually, it was a twisted item in this society, this isolated society, and this is what we believe in our society over the last 67 years. So how did they do it? Well, they actually use a combination of two plants, and so there's an interesting pharmacological issue here. The first plant contained this compound, which is actually made in our brain in small amounts. And it's rapidly degraded, and so normally there's very, very little of it, but it is a hallucinatory compound, so it causes hallucinations. So if they were to take this compound by mouth, as it was absorbed through the intestinal wall, the compound is going to be degraded by monoamine oxidase. And so that evolution of the survivors of the early days before we knew any of this stuff, we selected for people who could do very good metabolism in the intestinal wall, and hence prevent compounds getting into the bloodstream that otherwise would cause toxicity or other things. And so if they took it by mouth, which is the way they make a mash of the plant and consume this orally, so it shouldn't have any activity, except that they found another plant that if they mixed the extract of the other plant in with it, it contained this compound, and this compound is an inhibitor of the monoamine oxidase. So when you put the two together, then the first compound that should have otherwise been degraded is not degraded. And so now the shaman will go into a trance for 3 to 4 hours. And we'll get to this, I'll probably talk to you about GI growth later, but eventually that disrupts the process of intestinal motion and mixing, and the shaman vomits. And it's thought to be the high point of the entire process, when the shaman vomits. When he's got to that point, whatever he's thinking then is what the tribe is going to do afterwards. They're going to follow that line of thought. But it's a fascinating entity that they, without any of the concepts of modern science, had developed a way to make a drug more active. Now, in context of modern medicine, we call this drug-drug interaction. So sometimes when you are asked, or when you're going to dispense the drug, the doctor may ask you, what else are you taking? Because nowadays we've got a little bit smarter. We've realized that there are drugs that can't be put together because one will seriously enhance the effect of the other and take it from the normal range into a range that might cause toxicity. Now there are also people who have different levels of the metabolizing enzymes. And so that also becomes, that's something that you hear about in terms of personalized medicine. So it's to know ahead of time, what is the likely dose that we should give to an individual patient? Because at the moment it's a standard dose. And so, it's 2 Tylenols. But maybe for that person, 1 would be sufficient. Certainly my wife, she's very little. So she usually takes most of the tablets and cuts them in half. On the other hand, a lineman for the Alabama team or the Auburn team, might need 2 or 3 or 4 in order to get the same benefit, because they're different size. Anyway, so it's very, very interesting, figuring out what exactly what these people were doing and why. So this is, I regard this as a very important teaching point of how the metabolism of therapeutic can be dramatically influenced by a second drug, or even a non-therapeutic agent, something maybe not even thought about. When I got into this, I had a botanical center here at UAB for about 10 years, and we were looking at the uses of dietary supplements of various kinds. And I have to say that the School of Medicine at the time just didn't regard this as good science. But it was a very interesting supporting study. So the people were in a clinical trial. They were asked to study this drug that affected calcium activated events in hypertension. And they were given the option of drinking it with water, orange juice, or grapefruit juice. And when they did the study, after a while, they realized that the patients who were taking grapefruit juice were having much higher levels of the drug. So simple things like grapefruit juice that contain flavonoids can alter the metabolism of other compounds, and particularly the therapeutics. So now you have to be much more careful when you're using drugs to understand the background of the things that the patient is doing. And many times if it's a serious drug, they'll actually ask you, are you taking any dietary supplements? So we've moved ahead over the last 15 years or so. Okay, so we were sort of moving along in a very random way. In the 1800s, there was quite a lot of effort put into understanding the use of plants in what was to become pharmacology. And there were groups of universities that specialized in this, and these people were called the eclectics. And eventually the eclectics are going to be replaced by the modern medical school and the development of pharmaceutical drugs as we know them today. But of course the plant issue is still there. It didn't go away. So the arrival of synthetic organic chemistry that became available in the late 1800s, and then of course into the early parts of the 20th century, allowed us now to synthesize compounds as opposed to just extract them out of plants. And we still use material that's coming from plants. That's still there. And extracts from other things that are not plants. And then modern pharmacology, the concept of the drug having a target, began to become involved, and therefore the combination of these two developed pharmacology largely as we know it now. So some of them have an extremely old history. People have known for a long time that if you sucked on willow bark, it was a good method of getting rid of pain. And that knowledge going back 2,000 years. Well, the active agent in here is aspirin. So aspirin is actually a natural compound found in this particular tree. But the synthetic chemist now could actually make that compound in the laboratory, and therefore you didn't have to cut down trees in order to get the compound that was needed. And also, particularly in the last sort of 50, 60 years, the types of analytical methods we have now allows us to isolate the bioactive that might be in the plant. And once we know what that compound is, then the option is, can we make it more easily than extracting it from the plant? And what we're seeing nowadays is for very complex molecules, people are getting bacteria to actually put genes in bacteria, and bacteria can make very, very complex chemistries that couldn't possibly made by standard chemistry in the lab. We'll mention that a little bit later. So the idea of pharmacology goes back actually a long way. The Chinese, Greek, Indian, Japanese, Muslim societies have, in a way, had a precursor, even though they didn't have the right words, and they obviously couldn't isolate what a receptor or concept of a receptor. But they knew that there was a systematic way of applying compounds to human beings that could change the outcome of whatever disease they were experiencing, or infection, particularly infections. So as we understood the physiology of the human body better, then these new things and the chemicals associated with them can be put together, and that allowed pharmacology to occur. My first wife was an expert in pharmacognosy. And pharmacognosy is not pharmacology. Some people get that confused. So cognosy is the study of the plants that give rise to pharmacological compounds. So these type of people will look at a powder and put it under a microscope and tell you whether it actually came from a particular plant or not. They can that's their level of expertise. So pharmacology is all about understanding what the bioactive substance is that gives rise to the effect, and then the development of compounds derived from these bioactive substances that enable them to be used more optimally and to maximize the benefit. So there are three gentlemen, I'll move through this quickly, that really represent the pillars of the start of pharmacology. Rudolf Buchheim developed an institute, or founded an institute of pharmacology in 1847. Oswald Schmeideberg showed that muscarine, an alkaloid obtained from a plant, could have the same effect on the heart as electrical stimulation. So the idea that there was a connection between the electrical stimulation and a compound that was derived could actually cause the same effect was established. And then Paul Ehrlich brought to us the concept of chemotherapy and the idea that you could generate a magic bullet. In other words, a compound that would be targeted to a particular site. That could be to a receptor, to an enzyme, to a transporter. But the idea of having a compound that uniquely hits one place and allegedly doesn't hit anywhere else at the dose levels that are used. In order for this to happen, modern pharmacology had to have legislation, and this gradually evolved through the first 3 or 4 decades of the 20th century. So the establishment of the Food and Drugs Act, the FDA, and the Food, Drug and Cosmetic Act. And this created a way for the pharmaceutical industry to become developed, the use of patenting of the compounds. But of course it also came with the legislation, the need to ensure that it wasn't toxic to the other people. So the compounds that were being made by the pharmaceutical industry are compounds that are not present in the living world. They are unique compounds. And of course, I'm sure you've read, as individual drugs come up, there is a considerable oversight from the FDA. So when you develop a drug, you might do it in some form of cell assay initially, then you take it to an animal model, dog, cat, primate, and then to a human. And this is very expensive. It used to be get a drug to market relatively cheaply, but now it's probably several $1 billion between the initiation of the search for a drug to act on a particular event and the final delivery of something that the FDA approves of. And of course, this means a lot of money has to be obtained from you and me if we're going to buy that drug. And the companies want to ensure that they have the patent for enough time that they can recoup the costs that they have to spend in the first place. So it makes the pharmaceutical industry a major entity in modern society. And it's a constant problem, as we're seeing as the different forms of government go through about how to deliver medical care. So the cost of drugs is a very serious event. Okay, so the initial ways of going about drug discovery was to utilize the new knowledge that was coming from our study of physiology. So, for instance, the pituitary hormones, it was realized that the pituitary had bioactive substances in them. And so the early days of discovering and isolating these compounds required the use of maybe 100,000 to 1 million different pituitaries. So that would be the starting. And they sort of mash that up and then begin to separate out using a variety of separation methods. And so this led to the realization that there was something called steroids, the pituitary hormones I just mentioned, and insulin. Insulin was isolated, I think, in 1921. The first steroid structure was allegedly got a Nobel Prize in 1927 and 28. But in fact, it was actually wrong. It took another few years before the crystallographers remedied that. Often the oral administration of the drug was ineffective because the gastrointestinal system, it has two characteristics. One, it has the stomach that's full of hydrochloric acid. So if the compounds are not stable with hydrochloric acid, then they're going to get destroyed right there. And then also the small intestine, the cells that line the walls of the small intestine are just looking for anything that's going to cause trouble. And it's going to metabolize it. So it either is inactive, or if absorbed, it's excreted straight back into the intestine again. So it never gets in the body. So we have to play games, pharmacological games, in order to overcome this. And so if we were to administer estradiol by mouth, it wouldn't work. Very, very hard to get estradiol. You have to go up maybe 500 times on the dose that you would have physiologically in order to get any effect. And I'll talk about the penicillins more in a minute. But the original penicillins could not be given by mouth. And I was 5 years old when I had a serious ear infection, and I had to get penicillin in my rear. There was this nurse who came with this monster baby. When you're 5 years old, everything is a little bit bigger, and so I had 3 injections a day in my bone, and she always seemed to put the needle right down to the bone itself. But that's what was available. It was another 10 years before the oral penicillins were developed. And so, particularly in terms of antibiotics, we've had a profound effect on the life of humans. In 1900, the average age of life was about 47 years, and now it's about 80. So we've done very well in that period. However, the bugs are striking back, the empire. So we've had to deal with AIDS. Each flu that comes around terrifies people. There was the biggest epidemic that ever occurred happened just after the First World War. And 40 million people died from what was called the Spanish flu. And they were all the young people. It wasn't the old people, it was the young people. And then in hospitals now we've got these drug resistance bacteria. It's a really serious issue. And so a lot of people come into hospital for one illness and go out and coughing because they became infected by the organism in the hospital. It's a real problem. So penicillin is sort of interesting. So who discovered penicillin? Was it Fleming? You think so? It wasn't Fleming. So I did my PhD with this guy. He was the chemist who actually worked the chemistry of penicillin out. And this was his boss, Howard Florey, and they got the Nobel Prize together with Fleming. But Fleming wasn't the discoverer. Everyone uses the term discoverer. He wasn't the discoverer. Several people discovered penicillin. He happened to be the one who was alive when the Nobel Prize was given. Okay, so you can't give the Nobel Prize to a dead person. So the person who we probably should give the credit to is this guy, Ernest Duchesne, Frenchman. He was a cavalry guy. He was in medical school. He noticed that when he went into the stables, that the people who were looking after his horses would always put the saddle in a very dark part of the barn, and this green mold grew over the saddle. And he asked them, "Why did you do this?" They said, "Because the horses don't get back sores." So he took the green mold back to the lab, did experiments, showed that it killed bacteria, submitted it as a thesis to the French medical authorities, and had it rejected. So in 1952, with the Nobel Prize having been given out, the French government somewhat belatedly gave a prize to him posthumously. But actually another guy had noted the same thing. So this was John Tyndall speaking at the Royal Society, noticed that penicillium causes bacteria to burst. And then of course you have to think about the people who are looking after the horses. How long have they known about it? probably a lot longer than any of the people did. So, it turns out Fleming, if you go to Imperial College in London, the chemistry building is named the Sir Alexander Fleming building, and sitting right next door is the biochemistry building, which is the Sir Ernst Chain building. So these two guys basically didn't like each other, and the irony is that they are lying in the ground, as it were, right next to each other. We find that quite ironic. So, what was going on in penicillin? This is penicillin, and this is the four-membered ring, the beta-lactam ring. And that was extremely difficult to synthesize. Chain said in his Nobel speech that the chemists have all worked on this, and they just simply can't make this. So penicillin was acid and alkali labile, but it was recovered largely unmetabolized. If it was given it intramuscularly, you could collect the urine and you could recycle the urine and recover the penicillin from it, and then use the penicillin again. And there were drugs that slowed down the urinary excretion, so it kept the concentration there. So the real idea would be if you could make this compound, which is this minus the side chain, because then you could attach to this group almost anything you want. So Chain was very interested in, as part of the development during the Second World War of large-scale production of penicillin, in large-scale fermentation. And so a company in England asked him to do consulting work for them in the late 50s. And they used penicillin as their sort of compound to work with. And they realized that the two groups were measuring different levels of penicillin. And so much more in the group in England. And when the two groups got together, Chain was still using the killing reaction, but the company was, all of that was too slow, so they discovered penicillin gave rise to a color, so it had a colorimetric assay. So they were measuring something besides the penicillin that was in the broth of the penicillin mold. And it turned out to be the very thing they couldn't make. And so once they knew that, then they could go on and make all the synthetic derivatives, things like ampicillin and other oral penicillin. So we now have oral penicillins, and people don't have to be injected in the rum anymore. Okay. There's a worry that the age of antibiotics might even be over. The ability to make new antibiotics has really been slowed down. So people are getting quite concerned about this. And so lots of antibiotic-resistant organisms are coming along. And a lot of this has come from the deliberate overuse of antibiotics. So in the farming industry, it's possible to get cattle, chickens, and other animals to grow much faster if you put them on antibiotics. And it's something used there for infected things. Another form of bioactives is that in chocolate, the plant, not with all the milk and sugar added to it, is actually very bioactive. It was well known to the Mayans and Aztecs. And much like the shaman, this was used to improve alertness and energy. It was given to soldiers in battle, but only normally restricted to the higher ups in the society. So the Linnaeus decided that it was the food of the gods. And so it got mixed with sugar and fat, and you know, it was your Mars bar or whatever thing you like. Anyway, another colleague who's from Tufts was down in the isthmus of Panama doing bird watching, and he noticed that the people there were having drinks laced with salt. He was very much interested in hypertension. He was convinced that they would have very high blood pressure. So the next year he came back with a meter to measure their blood pressure and discovered it was low. And also there was no change in blood pressure with aging amongst the people there. So he realized there had to be something they were doing, and it turned out to be consuming this drink, not the chocolate, but the drink. And he took it back to Boston, gave it to patients. and found that it was able to lower the blood pressure of these patients. And so what it was doing was affecting the glomerular filtration of the kidney, and therefore they were able to excrete a lot more sodium. The sodium load could be higher without consequence to the blood pressure. So there's lots of bioactives around, they surround us. Okay, so let's get down now to talking about some of the words that you're going to take away and when you understand the concept of pharmacology. So we've said the targets can be receptors, enzymes. These were not known until relatively recently because we didn't have methods for isolating them or sequencing. And so some of the compounds were developed long before we had the ability to study them directly in the chemical nature. So there was an alkali called muscarine, which targets the muscarinic receptor, and I'll talk about that later in the course. Adrenaline, the adrenal receptor, and it turns out there are two of them. Acetylcholine also interacts with the muscarinic receptor, and the second set of receptors called the nicotinic receptors. There are histamine receptors. Turns out with pharmacology development, there are two of them. One you might take if you were driving in a car and you were prone to getting sick, and another one if you had an ulcer. So the H2 receptors are used for ulcers. And then estradiol, there was the original discovery of estrogen receptor alpha, and then later we discovered another completely different receptor on another chromosome that was also responding to estradiol. So these things, gradually emerged as the science got better. So the target, we can be enzymes, we can have both inhibitors or activators. So sometimes the compound actually increases the activity of the enzyme. Many times it's an inhibitor. It can be a receptor in the membrane, like the insulin receptor, or it can be within the cell, intracellular, like the estrogen receptor. It can be ion channels. Obviously that controls things that are going in and out of the cell through the natural holes. And also transporters, so things that bring in glucose, amino acids. And then we worry about drugs that are trying to pump out foreign compounds. So this becomes very important in cancer because there's a large amount of multi-drug-resistant transport. And this is probably too complex, but this is just a system with a receptor sitting in the membrane. There are drugs that can activate the receptor in the membrane, bring two individual receptors, molecules, bring them together. When that happens, they then autophosphorylate themselves. So you can have compounds that inhibit ligand binding to start this process. You can stop the phosphorylation, and you can also stop any of these downstream effects that eventually will lead to expression of genes in the nucleus of the cell. So many places in which the compound can have as its target. So extracellularly, close to the membrane, and then intracellularly. So talking about now, how does a drug interact with the target? Okay, so it's very important that it has the right shape. So let's take a very simple example here. So here's a compound. When it's applied to the cell, it causes the receptor to start having other proteins associated with it. So it's building a complex. But if we now put this compound in, the receptor now can no longer hold on to the proteins that were bound to it, and therefore the receptor complex dissociates, so you don't get the benefit of having the organization that otherwise was occurring. So we can think of the receptor of being in two states, a state where it's inactive and a state where it's active. And so things move between these states. So here's the state in the system before we do anything. So it's called the constitutive state. And then we have the state that's activated by an agonist, in other words, a compound that causes an effect. So drugs can either bind to the receptor and increase its activity, so we call these agonists, or they can bind to the receptor and prevent its transfer to the active state, and these are called antagonists. You know, where I put blue words here, that's just sort of hint of things that you really have to understand, because these will be things that will go in the exam. And so there are not only binding occurring in the active site. So we've got two sites here. One, this is the active part of the protein's activity. So an agonist can go in there, and also a competitive inhibitor can go in there. But you can also have compounds that are allosteric, and they bind at another place on the molecule that causes a shape change. But once these bind to the molecule, the whole molecule undergoes a shape change, changes the size of this region, and now the agonist and antagonist can't work anymore. So what you might see is this is where you have a compound where you're looking at, this is the normal activation. So this is the dose increasing this way, and here's the response going up here. You can have a competitive inhibitor, and it will do this. Eventually, since it's competitive, there is a concentration of the agonist that will still work, because the two are competing for each other. So if the amount of the agonist exceeds the amount of the inhibitor, the system finally responds, but only at a much higher concentration of the agonist. If something is a really good inhibitor, then it can never reach the maximum dose, or maximum response. So this one is a true inhibitor. And there are actually compounds where the binding to the allosteric site can actually improve the structure of the active site. And there, when you have those, you actually can enhance the overall effect that you would get with the normal agonist on its own. So this is agonist activation, and this is antagonist inactivation. Okay. So you can have other things go on. So this is the background, the constitutive activity. So if you have a full agonist, it will eventually cause a maximum effect at some particular dose. You can have a compound that we call a partial agonist that will allow increase in the activity. It will never reach the level that the full agonist does, no matter what concentration you use. You can have an antagonist that prevents the activation, but you retain the constitutive activity. And then you can have what they call an inverse agonist, where you can actually inhibit even the constitutive activity as well. And so this would go down. And I think my word is full. So we talk about full agonist. This is a full agonist. A partial agonist. A full antagonist. And an inverse agonist. We actually inhibit both. So what is the drug? The drug obviously has to be complementary to the site to which it's binding. So you want the two have to be sort of mirror images in a way of each other. So here's an empty receptor, and if we have a drug that's round about the same shape as the hole in this receptor, then you get binding and you see agonist activity. But in this case, the drug we've applied is a square, and it simply won't fit in that site. So the shape of the drug is very important as to how it fits into the shape of the receptor, whether it be an enzyme, a true receptor, or a transporter, or an ion channel. So there has to be considerable likeness between the two, both in size and also in the chemical nature because there's going to be a lot of chemical interactions that are going on. So sometimes compounds can actually covalently bind. So they not only come up to the receptor, but when they get there, they actually chemically form a bond. And we'll talk about one of those in GI drugs. It's a drug called omeprazole that actually forms a chemical bond with the receptor. Most of the rest are non-covalent, and that can be driven by electrostatic charge. So an ammonium group, a carboxylic acid group, a sulfate group. It also can be due to hydrogen bonding, where there's some small charges on an amino group or hydroxyl group. It can be aromatic rings coming together, so the aromatic amino acids and the aromatic drugs will like to get together. And then there is also forces that start to become important when drugs get very, very close to each other, called the van der Waals effects, and these can be extremely strong. And then we have to think about the fact that they not only have got to have all these things, but they've got to be in the right mirror image. Because if they're not in the right mirror image, they won't be able to come together and get the maximum interaction between the target and the drug. And then, so a lot of the natural compounds are very complex chemically. So they have, if you've ever done chirality in chemistry, you probably didn't want to remember it. And it's a real issue. It's extremely hard to control, even with modern chemical technology. And so people have tried to avoid using compounds with chiral centers. But this is shown to be very limiting. So we're now going to put, or people are putting into bacterial systems the entire synthesis that was present in a plant, and now make it work in bacteria, and actually drive up the yield much, much higher. So this is an example of a compound. This is Taxol. It's used as an anti-cancer agent, got lots of chiral groups on it. This is oxidosqualene, if you remember the synthesis of cholesterol. This is a hydrocarbon that is in one step converted to this compound. It's the first sterol. There are 1,638 possible isomers that could be made, but nature only makes one. So it's much easier for nature to make it than for a chemist. As I've said several times, the challenge is getting is just selecting the right drug and at the right dose. And we should have a wide therapeutic index. So a Tylenol, for instance. Most people would take a couple of Tylenols if they had a fever. Yeah. But if you take too many, and too many is 20, you can actually turn the liver into a failure situation, and people will die from it. I worked in a liver unit in London, and about half the people who came in with a Tylenol overdose went out in the box. So it's because they displaced good fine. So combinations of over-the-counter drugs, people, particularly actors, sometimes people come back from fighting in different parts of the world, come back with chronic injuries, and they go and self-medicate. And even the self-medicated over-the-counter drugs, if you take enough of them, many combinations can be lethal. So Paracelsus is right. It's all about those. Okay, I'm getting the hint that we should take a break now, so let's stop at this point, and we'll pick it up in